So here we're looking at the beginning of meiosis one. And what you can see in prophase one is that the maternal, which means from your mom, and the paternal chromosomes, which means from your dad, they're joined together to form this tetrad. Now, when the tetrad is formed, the non-sister chromatids, so for example, the chromatid from mom and the chromatid from dad, they can actually join together at a part called the chiasma, and they can actually trade places. And this is one of the main ways that we mix up our DNA before sexual reproduction. So at the end of this, what we have here is some of our chromatid are now swapped. We've swapped parts of our DNA molecule from here and parts from here. And this is why the concept of homologous chromosomes is so important. You have to understand that these two chromosomes are homologous, which means they have the same type of information. And that's why it's okay to swap their pieces. If one of the chromosomes had something about hair color and the other one had something about the size of your heart, you couldn't just trade the DNA because you might be missing some really important information. The only things that get traded are things that are the same trait. So you're just trading one hair color gene for another. You're not trading two different types of information. It's just one type of information. So now, when we split these up at the end of meiosis 2, you can see that some of the chromosomes are similar, like this one still looks like the paternal, and this one still looks like the maternal, but these ones are recombinant, which means that they are a new combination of DNA. It's half, it's mixed up. Your mom and dad's DNA gets mixed up, but not for every single chromatid. So when they finally get separated at the end of meiosis 2, you can see that some of your gametes have just a purely maternal chromosome that's exactly like the one you got from your mom, but some of them are mixed up, half mom, half dad. The mixed up ones would be called the recombinant chromosomes, and the ones that stay the same are called parental chromosomes. This can be kind of a hard concept, but there's plenty of videos online. And I think if you read through the chapter and still are feeling confused, definitely come talk to me about it and I can help you with this. So the last section that we're going to get into is what can happen if there are mistakes with either the number of chromosomes or if the chromosomes don't go to the right place or if they don't get divided correctly. So we're going to kind of look at all of the other cases of like things that could happen. So this is kind of just the last cases, the exceptions, I guess. So an abnormal chromosome count is the result of non-disjunction, which means that basically the homologous chromosomes do not separate correctly during meiosis 1 when we split up the tetrad, or the chromatids don't separate correctly during meiosis 2, the end of meiosis 2, when everything's supposed to be splitting up and going into the gametes. So either of those count as non-disjunction. And junction means joining, disjunction means breaking, and non-disjunction means not breaking. So non-disjunction means the chromosomes are not splitting apart correctly. For example, what they should do at the end of anaphase 1 is this tetrad should split into two separate chromosomes, but in non-disjunction, they didn't split apart. And the result of this is that once we get to meiosis 2, we have one cell with way too much DNA and one cell with not enough DNA. So this is a problem. These would be abnormal gametes.